Okay, welcome everyone to the Diversity in Biology seminar series. We would like to start by acknowledging the ancestral territories of those on whose land we are currently living and working. We anticipate that since this is a UBC Okanagan event, many in attendance are in Kelowna on the ancestral and unceded territories of the Syilx Okanagan Nation. Many of you are joining us from other regions, including our speaker, so we encourage you to reflect on the lands on which you live and work and who the ancestral caretakers of the land might be. With that said, I would like to introduce Dr. Mehran Dastmalchi, my colleague and friend from the Department of Plant Science at McGill University. Mehran received his PhD from Western University and then completed 2 postdoctoral fellowships, one in Professor Peter Ficini of University of Calgary, and the other one in Dr. Vincent DeLuca of Brock University, both international authorities in the field of plant natural product metabolism. Mehran then joined McGill University as an assistant professor in 2020. And he is currently setting up a research program to study metabolism in specialized metabolism in legumes with the interest in pathways producing defense and health-related compounds. The title of his presentation is Looking for Unknowns in Specialized Metabolism. Dr. Dastmalchi, you have the mic.
Perfect. Thank you, Sohail. Very kind introduction. And thank you for inviting me to speak to the UBC audience, I guess. And thanks everyone for being here today on what feels like the first day of spring here in Montreal. And we're finally thawing and coming out from underneath a heap of snow. I will share my screen now. Wonderful. So yeah, as Dr. Mahmoud mentioned, I am going to talk about specialized metabolism today and a little bit about some projects that I've done in the past and looking forward as well. Before I start, I want to thank the people who have helped me get to this point. All right, so at this juncture in my career, I often reflect on who's helped me along the way and how I can reciprocate and also help the students that I come across and take all of the maybe the best features of my sponsors and mentors and try to replicate or simulate that. So I should first start with Dr. Sangeeta Dalbadel. She's a research scientist at Agriculture and Agri-Food Canada and adjunct professor at Western University. She was my master's and PhD supervisor back in 2010 when I started and I was applying for grad school. My application was in a heap of other applications. I was kind of in that anonymous pile that us faculty members kind of look at and think, well, there can't be any good students in there. And she took a chance on me, interviewed me, and then gave me a position in her lab. And beyond that, she was always there when I was struggling, whether it was with the qPCR or cloning or whatnot. And her door was always open for troubleshooting discussions. So I have to thank Sangeeta first and foremost. And my co-supervisor, this list of individuals here is kind of like the who's who of plant specialized metabolism in Canada. And Dr. Bernards is another prominent figure there. And he is an incredibly kind and insightful human who was there at some of the toughest junctures. Some of you grad students know that, a PhD is a long period and you have your complement of lows and highs. And Mark was definitely there for me during that process. Oh, sorry. I should also add, I want to thank Dr. Peter Ficini, who was my first postdoctoral supervisor. He gave me a real platform to pursue my interests, pursue them under the framework of morphine biosynthesis, but also work with industrial partners and work as as a person who brings ideas forward and executes them. So Peter was incredibly important. Finally, last but not least, Dr. DeLuca, whose lab I was in for maybe just about a year, but it's a testament to the magnitude of his character and his legacy in plant specialized metabolism that I think this was a crucial period for me. It really helped me think about what I want to do for my own research. So I thank all of these people first and foremost. So as the title suggests, I'm going to talk about specialized metabolites, right? So a lot of you might know specialized or secondary metabolites are sort of the small molecules that we deem not necessary or essential for life. So they're basically compounds that are necessary for plant interactions in the environment, in defense, signaling, and whatnot. But if you knock out some of the pathways, it won't be lethal, essentially. You know, you can have knocked out anthocyanin pathway in strawberries and get white strawberries, but the strawberry still fruits and goes to seed and whatnot, and you don't disrupt life, essentially. So specialized metabolites are important physiologically for the plant in its defense and survival, but also they are restricted in time and space, right? So in particular, I'm interested in low abundance specialized metabolites. So this means that, you know, a specific cell type produces them, a specific developmental stage is key to their accumulation or biosynthesis. And that is what makes a specialized metabolite special. The de novo chemical synthesis of a lot of these compounds that we might be interested in for their bioactivity is environmentally and financially quite costly usually. This is usually due to their stereochemistry or complex nature, and they require harsh chemicals or physical pressure, etc., that makes de novo or complete chemical synthesis quite expensive. And as I alluded to before, they're low abundance, so cultivation in the plant and extraction from complex plant chemical cocktails is quite convoluted and arduous. That's why there's the burgeoning kind of field of synthetic biology and the whole promise of taking these plant pathways and conferring that competence to small synthetic engineered organisms such as baker's yeast or Saccharomyces cerevisiae, or perhaps algae, diatoms, etc. These solutions are often sought after because they're tractable. So that means you can do a lot of functional genomics, you can edit, you can change, they're scalable. You know, you can grow them in large vats, akin to brewing beer. And depending on the sugar source or the amino acid precursor, they can be sustainable. So depending on where we get the sugar from, harvesting it and providing it to the yeast, that simple compound can, through a long steps of metabolism, be changed into a complex compound. So that can also be quite helpful. It also allows us to go beyond the plant catalog. So say you have a compound that you extract from the leaves of a certain tropical plant, and you want to O-methylate it or hydroxylate it at a funky position that the plant doesn't do. If you have an enzyme from bacteria or somewhere else, maybe you can accomplish those tasks and create a new scaffold or a new derivative that goes basically beyond what the plant can accomplish. And the application or translation of this knowledge is in the form of taking these plant-derived or plant-inspired compounds and producing high-value products that can be used in medicine as nutritional supplements or in the agrochemical industry, which is a very large industry involved in stopping pathogen infection of plants, or basically weeds overtaking crop plant cultivation. So pesticides and herbicides. So there's a range of applications for compounds produced in engineered systems. And of course, this is just a small list of them. But before we engineer synthetic organisms, we have to understand how the plants transform these simple compounds to complex chemical structures. And that is usually called, elucidating the metabolic pathway, basically finding the genes that encode enzymes that take the small compound and take it from A all the way to Z. Right, so we want to find the genes that encode the enzymes and understand how the enzymes function. And that is easier said than done. And we still rely on the plants as they are the OG chemists. They have the ability, the enzymatic nature of these reactions to accomplish stuff that almost is entirely elusive even to synthetic chemists. And then we come to basically the major hiccup or bottleneck in synthetic bio. If you think of it as an IKEA catalog or manual, you open it up, you have some of the different parts, let's say the genes, and you try to assemble it together in yeast, and the pathway goes completely awry. You get products that you didn't expect, you get products that don't exist in the plant, and you don't get what you were hoping for. And it seems something is missing or broken. You can't call IKEA and figure that out. You can't, basically put in an order for the missing parts. And this is what I call the unknown unknowns. To kind of explain that simply, you're going from compound A to D over here. Let me get my laser pointer. You're going from compound A to D. You have 3 enzymes, E to 3, and basically you assemble them in yeast, and you're hoping to get compound D because that is the commercially valuable compound, or the bioactive compound. And strict synthetic biologists approach this as basically a circuit or different engineering elements that can be tuned, increase expression, lower expression, compartmentalize, etc. But that basically goes on the assumption that these are all individual, functioning alone, and also importantly, complete. They assume that this is complete. And one of the pet peeves that I have is that a lot of symbio papers don't even refer to the decades of plant research that has gone into finding these elements of often intricate pathways. So what happens often when you assemble these 3 enzymes in yeast and feed them compound A is that you get a compound you weren't expecting. And that's usually because there might be an auxiliary element that is missing, or something, whether it's enzymatic or basically an accessory or a transport or regulatory element. You are missing something. You're stripping these enzymes of their plant cellular context and hoping that it'll work exactly the same way in yeast, and it often, it rarely does. So here's a figure from a review article that I published last year. Here in particular, I'm looking at flavonoid metabolism, and I'm talking about a theoretical model where you have often a cytochrome P450 or an anchoring protein in plant metabolism interacting with soluble enzyme partners here in blue. And this is all tethered to the ER membrane. And this is going based off of my experiences with isoflavonoid metabolism, and the P450 there being isoflavone synthase, and it interacts with the soluble enzymes in the pathway. And so the intermediates are often forming local concentrated basically clouds, essentially, where they don't diffuse into the rest of the cell, and they go from partner to partner, passing between enzymes, and thereby precluding the formation or reducing the formation of byproducts, or what we would think as byproducts. And what we increasingly think is helping this process is this pink cloud-like protein that I'm envisioning here, which are auxiliary elements. I've looked in this review article at a couple of different examples of potential auxiliary elements. And this is the concept that I'm trying to think about and promote, is that, when we think about protein trafficking, when we think about the photosynthetic complexes, we always are aware, and through decades of research have come to understand the importance of auxiliary features that aid, guide flow or flux or the trafficking of proteins. But when it comes to plant metabolism or natural product synthesis, we just think of the enzymes and think, bim, bam, boom, you get the product. But that is rarely the case. There's a local molecular context to it that I think we are missing. So the first part, I am going back to a work I did in Peter's lab and talking about morphine biosynthesis and what happens ex situ, essentially, when you take morphine biosynthesis out of the plant and hope to replicate it in yeast. So when I started in 2015 in Peter's lab, people thought that the morphine biosynthesis pathway was over and done with. And 3 years later, we had, as a lab, discovered a couple of new enzymes in the pathway, as well as a transporter or a set of transporters. So what I believe now, my current bias, is that plant biosynthetic pathways are never over. So briefly about opium poppy. So opium poppy is a medicinal plant that has been in use since before the dawn of civilization. 5000 BC, you go to the Neolithic age, the humans in the Alps, in their little pouch of basically trail mix in the mountains, they had some wheat, barley. They also had opium poppy. We don't know what they were doing with it, but we have a strong guess. So opium poppy is the source of the precursor thebaine that naturally in the poppy produces codeine and morphine, and is used commercially to produce them as well, and also semi-synthetic opioids such as hydrocodone, oxycodone, etc. So to this day, we rely entirely on opium poppy for the majority of our opioids. And these are, of course, important compounds. They're important because they are major analgesics, so pain relief, both acute and chronic. And a lot of functions in society for humans would be impossible without opioids. You think of something as innocuous, maybe as childbirth, when a mother receives an epidural. What's in that epidural? Opioids. You break your arm, again, you need opioids. Of course, there is a major overprescription of these opioids, and by certain pharmaceutical companies. Of course, there was overselling of their qualities and underselling of their addictive properties. Okay, and of course, and now the black market is overwhelmed with extremely strong opioids that would be enough to tranquilize an elephant, and in tiny trace amounts can lead to overdose death. And during the pandemic, it's important to note that there were 20 deaths a day in the first 3 months of the COVID-19 pandemic due to overdose death. This is the unseen, invisible pandemic that, of course, we are acutely aware in Ontario, Quebec, and in BC, right? So the complete chemical synthesis of these compounds, as I've alluded to before, is tough because of the stereochemistry, and we rely entirely on plant cultivation, which is limited to a handful of countries and can make opioids quite costly, and is open to the vagaries of environmental disasters or a drought, etc. And it is very hard to predict the global demand for the opioid precursor thebaine, and therefore plant and cultivate the requisite amount of poppy. So this is a bit of a problem. So in 2015, the so-called last enzyme in morphine biosynthesis was discovered, and as such represented a big potential for synthetic biology to solve that problem that I mentioned, to be able to take all of the enzymes from the plant, transport it, basically transplant it into yeast and produce something like morphine. And there was a paper that did it. It was published in Nature, and Nature did a bit of a misleading spin and wrote a little puff piece named homebrew heroin, the possibility for this yeast strain to basically enable people to brew heroin or opioid products at home, which is farcical, to say the least. Mostly because the paper basically achieved a minute, minuscule, almost imperceptible amount of morphine. And thus the problem that I've mentioned before, this massive inability of synthetic biology to replicate what the plant does. As I've mentioned, there's only a handful of countries that grow opium poppy legally. 50% of the world's opium poppy for commercial use and for cultivation and extraction of thebaine, the precursor for those opioids, is grown in Tasmania, which is this island off the coast of Australia, which you might also recognize Tasmania as producing these wonderful boots, the Blundstones. So two things, two exports of Tasmania, poppies and Blundstones. Now, these poppies that you see in this field over here are accumulating large amounts of thebaine because they do not express the enzyme that basically metabolizes them down towards morphine. That enzyme is T6-ODM, which catalyzes an OD methylation at the 6th position to form this compound called neopanone. You'll hear me talk about this a lot today. Neopanone occurs in aqueous solution in a 3-to-1 equilibrium, favoring this other compound, codenone, and you see the double bond has shifted over here. And it was thought that perhaps the same thing is happening in the plant. Thebaine is being OD methylated, and neopanone is basically been shifted to codenone, whereupon this enzyme codenone reductase, which is NADPH dependent, is reducing this keto group to a hydroxyl. And it was thought here back in 1993 and until recently that core was selectively reducing codenone to codeine, okay, and was not touching this other compound, neopanone. And thus it was using up the substrate and shifting the equilibrium until nothing was left, and basically you produce codeine. And finally, at the 3 position, it is OD methylated by another enzyme called CODM to get morphine. So you go from thebaine, neopanone, codenone, codeine, and then morphine. 3 enzymes, 1 spontaneous reaction. And so this group at Stanford took these 2 plant enzymes, introduced them into yeast to produce codeine. 2 enzymes, simple. What happened is that they got a new product called neopene at the expense of codeine. And here you see that double bond is shifted as with respect to codeine. And it accumulates at the expense of codeine. Here you see there's more neopene in the yeast extract than codeine. And this represents a problem. You're trying to get one compound and you are ending up with its isomer. Furthermore, rather than getting morphine, they were getting O-demethylation, perhaps of neoprene to neomorphine at the expense of morphine. So this represents a major byproduct pathway and a major problem. Now the question is, does opium poppy accumulate either of these metabolites? Or is it an artifact of the enzymes being expressed in yeast? Or is the reduction of neopenone to neopene inherent to codenone reductase, contrary to previous reports? Now, I basically started out in the lab and I did a range of different assays on core or codenone reductase. Here, I'm going to briefly go through the major outcome, right? So I incubated a core in the reverse reaction, feeding it codeine and providing it NADP. That gave me, in a 3-step process in the same tube, neopene. And how is that happening? Basically, core is producing codenone and also NADPH. And this spontaneous reaction is occurring, so neopenone is being produced. And core is then taking the NADPH it produced itself and reducing neopenone to neopene. So you're getting basically this isomerization of codeine to neopene in one single tube in a 3-step process. I tested a range of different cores from different varieties of opium poppy. And I found one variant, one isoform of core from ornamental opium poppy that I named core B, which was far superior in the general reduction or oxidation reaction as compared to any of the other cores. So this new isoform was the most efficient catalyst and furthermore had thermo and pH stability that was incomparable to the others. But the main problem was neopene. So if you incubate core with neopene and provided NADP, what do you get, right? Do you get neopenone? And the answer is no. So neopene is not accepted as a substrate by core. So this reversible arrow is not reversible, and indeed it only goes in one direction, and you accumulate neopene. And the same goes for the parallel reaction of neomorphinone to neomorphine catalyzed by core. It too is irreversible. So these two metabolites are enzymatically trapped. Once you form them, you can't go back. Here, going back to that superior core B, we also did some crystallography and some work and found 4 key residues that were linked to its superior function. One of them being this valine. So the alanine mutating to a valine, bringing it closer to the NADPH cofactor and improving the output by 20-fold. But the problem here is that you're producing neopene, even if your core is superior and functions really rapidly. What you're getting is just even more neopene, which is not what you want. So we need a mechanism to curb or preclude the formation of neopene. And so synthetic biologists did a bunch of different acrobatics to try and fix this, different compartmentalization, different levels of expression, and none of that worked. The fact of the matter is that if you put core into yeast and feed it codenone, codenone is automatically going to basically form this 3-to-1 ratio of codenone and neopenone. Some of it gets reduced to codeine fast in the faster forward reaction, but over time this is going to flow back, and you're going to get the reduction of neopenone to neopene. And because it's enzymatically trapped, as this arrow is not reversible, you get accumulation of neopene, and that is a major problem. And when we tested it and put T6-ODM and core into yeast and fed it thebaine, we got basically accumulation of neopene at the expense of our desired product codeine, further corroborating the previous work. And when I put my superior core B in there, we got increase, 10-fold increase in overall output, but the major bar, this black bar, is the neoprene, and that is still a problem. So you get better output, but you're still getting neoprene. So how do you fix that? So we basically went on the hunt for a catalyst or a protein feature that would solve this problem. So often in synthetic biology, when new products are formed at the expense of your desired product, it's called carbon loss. And so I was working in Calgary, so borrowing basically a pipeline metaphor, there's a leak in the pipeline and you want to fix that. So we were looking for a novel component. So the first question is this reaction between neopenone isomerizing to codenone, is that spontaneous? And I'm talking about this unsaturated bond shifting from the C8, C14 position to the C8, C7, right? So I'm talking about this double bond shift. Does that happen spontaneously in the plant? So here is the opium poppy pod. And if you use a razor blade and cut it radially, you get the oozing out of the latex, which is the cytoplasm of the lenticifer cells of the opium poppy pod. And the latex accumulates the right stuff. So by some unknown mechanism, it was precluding byproduct formation. You mostly get codeine, very little neoprene, and you entirely get morphine and no neomorphine. So how is the plant doing that? As I mentioned, the latex is where all of this stuff happens. All the latter stages of morphine biosynthesis, both the enzymes and the alkaloids corresponding to their function accumulate in the latex. So if you're looking for a mechanism that would preclude byproduct formation, our best guess was that it would be in the latex. So we basically had an inkling that the latex would have this family of proteins called pathogenesis-related 10 proteins that might be candidates to solve this problem. And the reason was because we had recently discovered another basically enzyme that catalyzed a spontaneous reaction, and that was called thebaine synthase. And that was a PR10 that we had recently found that was also enriched in the latex. So we basically started out doing some omics, looking at the proteome and transcriptome of the latex as compared to the stem. And we found 15 PR10s enriched in the latex and 18 using transcriptomics. So this gave us a good sense of potential candidates that are co-expressed with the latter stages of morphine biosynthesis. So I expressed all of these different PR10s in E. coli, purified them, and conducted assays coupled with core. Now, why does it have to be coupled? Because we didn't think that this spontaneous reaction would be perceptible. Because as soon as you, let's say you incubate codenone with a potential isomerase here, whatever the result is, once you put that product in aqueous solution or you put it on the LCMS, you're going to get a 3-to-1 ratio of codenone neopenone. There is no physical way for us to get a completely pure neopenone standard or a completely pure codenone standard. So there's no direct way at testing out if there's a catalyst for this reaction. So we had to do coupled assays with codenone reductase. And what I did in this assay is incubate codenone, which is of course in a mixture with neopenone, with core and with different PR10 candidates. And we're hoping to see what the percentage of neoprene is as a percentage of the total product, which is codeine plus neoprene. So neoprene over codeine plus neoprene. And if that percentage is reduced, then we know we have a catalyst or a protein feature that is pushing this reaction towards codeine. So codenone plus core or core plus PR10. and measuring neoprene as a percentage of total product. So we tested out the different PR10s, and there was only one PR10, PR10-3, which reduced the neoprene produced as a percentage of the total reaction products. And we renamed this protein neopenone isomerase, suggesting that it was isomerizing this reaction from neopenone to codenone. Here is basically a theoretical model of a PR10 type protein, and you can see that it has these beta sheets over here with three alpha helices, and the C alpha helix basically comes over the beta sheets, and it has been called basically a baseball glove grip. And it has this large hydrophobic cavity, which has been known to either interact with lipids or other hydrophobic metabolites. And in this case, it basically is accepting this hydrophobic metabolite and potentially leading the isomerization from neopenone to codenone, or in effect binding this ligand or this intermediate and guiding flux towards codeine. And in other reactions that I did, I showed that NISO basically can push either this forward reaction towards codeine, or depending on a pH, an alkaline pH, push it towards neoprene, which is why I think it is more of a catalyst than an auxiliary feature. Now, if you take a NISO and you plop it into yeast, along with T6-ODM and core, basically going back to that 2015 paper, what does the inclusion of NISO do? So does it guide flux from thebane to codeine and preclude the formation of neoprene? That was what we were hoping to see. And indeed, the inclusion of NISO basically resulted in negligible amounts of neoprene and a 200-fold increase in the yield of codeine in yeast. So it guides flux. So if you are a synthetic biologist or you're trying to commercialize this pathway, you can't function without NISO, because NISO guarantees that you get the right stereochemistry, you get the right double bond in the right position, and you get the desired product. And without it, you go woefully awry. So NISO is necessary, and it was another unknown that was in the plant that no one was looking for, because this reaction was always deemed as being spontaneous, because we saw it as being spontaneous in aqueous solution. Now, as I said, you can go beyond the plant catalog. So if instead of core, you provide the yeast with this enzyme from Pseudomonas putida, morphinone reductase, or MORB, what happens is that you go and produce hydrocodone. And does the inclusion of NISO aid or guide that flux? Indeed, it does. So the addition of MORB increases the production of hydrocodone by 30-fold. And why is that? We no longer have core in this yeast module. We have T6-ODM, NISO, and MORB, but still you are precluding the formation of any byproduct by guiding the flux from neopenone to codenone. You're increasing the efficiency of this isomerization at a neutral or slightly acidic pH. Now, the next frontier of going beyond the plant catalog, which I assume would be pursued, is basically removing this methyl group off the N and trying to derivatize it further, including the production of the opioid antidote naloxone, which is a life-saving drug often marketed or sold as or provided as Narcan. And this can basically is known as the Lazarus drug, which can bring back opioid overdose victims from that experience, because it basically nudges out the opioids from the receptors. So that is basically the importance of looking again and again at the plant to see if we are missing different features. You know, sometimes you think you have all the catalytic function, but what if there are other enzymes aiding and abetting that function, guiding it, precluding the formation of byproducts? Anyone who has had the experience of taking plant enzymes and trying to put them into yeast knows that you are invariably going to get new to nature compounds. Sometimes those compounds are useful. More often than not, it's not what we're looking for. So it's important to continuously pursue plant specialized metabolism and looked at a plant for features that we might not have considered. And of course, an unbiased or untargeted approach to that is important. So I'll finally finish with some talk of what I'm doing in my lab right now at McGill. So I'm continuing a partnership with Dr. DeLuca, but today I'm going to talk about the main thrust of our work, which is looking at legumes. And I'm just going to give a brief overview. I only got my first two grad students last September, and they are working. They're working hard, but we are basically in the first nascent steps of establishing our research program. I just want to talk mainly about our goals. So as we know, of course, plants have a lot of different physical and chemical defenses, from thorns to toxins. And as I mentioned before, sometimes these toxins or defense compounds are localized and restricted to a specific tissue or cell type that confers some form of resistance, whether it is anticipating or induced by attack. One set of defense compounds are pterocarpans, which I'm returning to the field of flavonoid and isoflavonoid research, and that I pursued during my PhD. And these pterocarpans are a scaffold that is derived from isoflavones. So it basically has this additional ring over here. And they are characteristic of legumes, the Fabaceae family. And since I am at an agriculture campus, I am looking at forage legumes, including red clover, that are important commercially, as well as being important drivers of sustainability. So these pterocarpans can be potent antifungal and antimicrobial compounds, and they are often limited or induced in root tissue. So we have a particular interest in that. And one of my students is working on different elicitor assays to get those pathways induced and responding to either abiotic or biotic stresses. Here's that student. Her name is Fan Li, and she is working on space and time. So the spatial temporal profiling of isoflavonoid content in red clover. These are images that she's taken of different stages of leaf and flower development. Basically, we are going to look at different stages and tissue types, as well as eliciting function. And we're going to basically map the isoflavonoid profile of both the content and composition with keen interest in low abundance compounds. And finally, we're going to take a curated set of those tissues and launch an omics study. So that is currently what we are dealing with day to day. And as many of you probably know, that follows, metabolomics, transcriptomics, taking candidate genes that are co-expressed with metabolites of interest or known enzymes of interest, and cloning, protein expression, enzyme assays being the in vitro component, as well as in vivo silencing of genes in plants, as well as what we're trying to develop, which is multiplex cloning into yeast, culturing it for the major isoflavonoid scaffold, and then adding new components, new novel genes, and using yeast as a functional genomics platform to identify and characterize new enzymes, and hopefully get purified pterocarpans some day. So finally, I want to thank the members of my lab. So Jonathan was my first student. He was a summer student excellent student, decided to follow his passion of veterinary science, unfortunately for me, and he did some excellent work, and now that's being picked up by Fan, and my other student, Brandon, who is working more with cloning genes into yeast. I have to again thank Dr. DeLuca, Dr. Ficini, Dr. Dabadel, and collaborators here at McGill, Dr. Stefan Bayan, and our funders. Now, before I take questions, I should, I guess, direct us to Sohail. I have a couple of slides to, I guess, discuss the diversity component of this lecture. So I can maybe perhaps take questions on the science first, and then we can move on, or whatever the panel thinks would work best.
Sure. I think it's a good idea to address the science first. However, I'm going to refer this to Robin and see what she thinks.
I'm happy for you to keep. If you've got more slides, you can do it all at once, and then
Sure.
the questions that I've mixed together. But that seems to have worked thus far, so it's up to you. It's really up to you.
Excellent. Yeah, let's do that.
Okay.
So just 4 more slides. So I think of this of merit and, right? Recently, not recently, I mean, this is, we're sort of in the eye of the storm when it comes to culture wars, and also the concept and question of merit and how that interplays in science, in recruitment, in hiring. There was a fellow from McGill prominently in the news who was saying that he only hires based on merit. And this always kind of stifles conversation, and as well makes me think about what merit is, right? We think of scientists as if it's some sort of quantifiable thing. You can look on paper and say, this person has merit, this person doesn't have as much merit. So what does good look like? Is it quantifiable? Can we really ever say we hire purely based on merit, or is that, is that misguided? So early in a career, individuals are evaluated based on functional capability and expertise. The hard skills. Can they pipette? Can they grow plants? And these individual abilities of getting things done, right, in the lab. So that's how you might judge a grad student or a summer student to see if they should go on to do a PhD or not, or to hire a new grad student. But as one rises through the ranks, the concept of leadership skills, the concepts of suitability, likability become assessed, and there's a lot less clarity and alignment on what defines effective leadership and individual potential. How do we seek that, and how do we judge it? I've been on both sides of recruitment recently, right? And I find these are very, very subjective attributes to assess. So this question of merit always boggles my mind when people think it's a black and white situation, easy to decipher, easy to essentially quantify. And merit looks different at each of these stages, right? You go from the undergraduate all the way to being a member of a faculty. So what does it mean to have merit as an undergrad? So in white, I have the common notions, and in gold, we have some of the unsaid or unwritten criteria. So how fast can you read? It really becomes a problem when you have human physiology, plant physiology, you have 5 courses, constant material to read, and you're expected to remember that, either regurgitate or testing efficiency. How good are you at multiple choice? And you sit there an exam a couple hours, you have 80 multiple choice. Each multiple choice has beyond the A, B, C, and D, A, B, C, etc. You get the drift. And how good are you at selecting the program that fits you best? I remember being an undergrad and telling my mother that I was going to go study biochemistry. Well, you get to the University of Toronto, and the first year you're basically in life sciences, and then after that you have to basically earn your way into one of the specializations, and that is easier said than done. So navigating that, of course, is known, right? So that is the known stuff. What people don't tell you from the outset, and what I find when I teach undergrads now, is that a lot of them don't know what is lab experience, what happens in the lab. Sometimes they don't know that in this building where the teaching happens, there are also labs going on where people are working, basically 9-to-5 jobs. A lot of students don't know that they have access to summer internships, summer awards. And of course, those are all harder for international students. And if you either get you manage to get a USRA or some other summer experience, or maybe you do an honors thesis, what is going to elevate you and get you into the right lab for grad school is reference letters and sponsors. So people like myself now in this stage have to sponsor students to say, this person can get it done and would be a good student in your lab, right? So this is some of the unwritten stuff that the majority of undergrads that I see when I teach my intro plant bio course, they have no idea about the stuff in gold, right? They don't know that every summer that they're not gaining experience or working in a lab or something is a summer lost. Most of them don't have the privilege of earning next to nothing in a summer internship in a lab rather than working a job that actually pays them. That's the undergrad level. Then you get to grad school. Of course, you're told to work hard, persistence. Sometimes you work in a lab that allows you to be part of the ideation process. Execution, of course, is important. Publications is the albatross that hangs above all of us, right? And of course, there's the unsaid stuff. Some people are not great at growing plants. I have 2 students now. One of them is great. The other one never knows why his plants are yellow or whatnot. He says, maybe I'm not good at the plant stuff, but you need to encourage and cultivate that. And of course, having, being good at pipetting. And this is all stuff that you don't know when you go into grad school, whether you're good at it or not. Then comes the soft skills, right? That you either don't have time for, or you think that the time spent on it is time wasted. So working on your presentation skills. And again, stuff like likability, right? So that is determined by the status quo. That is determined by people at conferences, faculty members, people that are above you in this academic ladder, determining what likability is. The ability to network is also important, and really forcing your supervisor to send you to conferences. That is important. If you have your own means, of course, going to conferences can give you an edge up, or going to training workshops, etc. But again, I emphasize the last point here: mentors, important. Even more important, sponsor. And the difference between the two is subtle, but a sponsor, aside from guiding you, is also the person saying, hey, I have the student, so-and-so, they would be good in your lab, right? So many times I see these at conferences where the person is like, oh, are you Bill's student? Are you Bob's student, whatever? Oh, I know that person. So that must mean that you are good, because by the virtue of you being in their lab. And of course, if Bill or Bob is there saying, yeah, he's really good, Mehran is really good, that really helps you. That is more important than the publications even on your CV, or it can be, just to get your foot in the door. You get to the postdoc side. Point of your trajectory. And this is even harder. Now, the right lab, the right supervisor that you pick, it can basically determine your future. And this was an important part for me as well. And they tell you as a postdoc to focus purely on the work, right? And you need to focus on publications. And sometimes For example, for me, what worked well was Peter came to me with a couple of different projects where he said, this is low risk, this is medium risk, this is high risk. And you have the ability to pursue a couple of different options, and hopefully at least one of them gives you a paper rapidly. And we're talking rapidly, like within a year, you need to go from ideation to execution to publication. And that is incredibly tough. And then in the yellow is the stuff that people don't necessarily tell you. You need, at this point, when you finish your postdoc, to have a body of work, something that has a theme. You know, if you've worked in a completely different system or a different set of tools, and then you're doing something different in your postdoc, well, that might hinder your possibility of getting a faculty position. So you need something to be like, oh, this person has always worked on plant specialized metabolism in different systems. He's used different tools, but they've always, they've had a singular focus, right? And that is a privilege. You don't often come across, if you want to stay in Canada, 5 or 6 labs that meet all those requirements and are magically looking for postdocs at that time. And of course, while you're a postdoc, holed away in the lab, trying to accomplish all this stuff, you also have to be a likable, gregarious person who gets along well in conferences, and that comes easier to some rather than others. Then somehow, when I'm on the recruitment side, I'm looking at these people who are postdocs, they're somehow supposed to teach as well. You're supposed to have supervised people, either officially or unofficially, but also, do they have any experience teaching? Well, when is that supposed to happen, right? You have to carve out a little bit of time for that. And again, I was lucky that Peter allowed me to teach a couple sessions for his one of his courses. And then again, we come to sponsors, right? Your supervisor being your cheerleader to say, this person is good. And more important, unfortunately, is the pedigree of your supervisor. Are they known? Are they respected in the field? Unfortunate, but I find quite true in a lot of scenarios. And maybe I should add the caveat that this has been my experience and the experience of people I've spoken to. And there's definitely footnotes to all of these experiences. Finally, as a faculty, as a member of a faculty or a candidate to be a member of a faculty, you need to do a range of stuff that you have never done up till now: personnel management, finances, writing, not just grants, not just publications, but grants, service, representing on different committees, and teaching, taking up a massive amount of your time. And beyond that, the soft skills, again, of being collegial, getting along with people. And then still to this stage, as an early career researcher, you need sponsors. You need people like, for example, Sohail, who invites me to present at this in this seminar, people who invite you to write, etc. And here I want to give a little bit of an anecdote that is personal. I was invited last year to write for a significant journal, to write a focus review. And at that moment, it was a person that I knew collegially, who was a colleague, sorry, who invited me, and I thought, wow, this is a bit of a privilege. I don't know how I feel about this. Again, the question of merit. Did they go through a list of 100 people in specialized metabolism and say Mehran has the most amount of merit to write a focus review for a significant journal? That definitely did not happen. It is networking, and it is the fact that maybe I come across as being competent for what I'm talking about. But of course, there are a lot of competent people out there who didn't get that invitation. So merit, schmerit. I don't believe it. I don't buy it. But then the flip side of that happened. So I wrote a review. It goes to reviewers, and reviewer 2, hilariously, we always talk about reviewer 2. Reviewer 2 was an expert in the field who didn't believe I was in a position to talk about that subject, because they have made their entire career on that subject and don't believe that I was bringing anything new to that concept, which is interesting because, I try, if you read any of my reviews after this or publications, you'll see that I go to great lengths to add value, but they rejected it right offhand and said that basically it wouldn't pass, right? So I had to basically, and then beyond that, the main editor said, oh, this person's an expert on the field. I'm not even going to invite a third reviewer because that would offend them. So as an early career researcher, you have no ability to get past certain unwritten gatekeepers, essentially, comes to it. So that is that part of the spiel on sponsors. So this is my trajectory: University of Toronto Western, Calgary, Brock, and finally McGill. And the elements of luck or basically sponsors for me. At the University of Toronto, I got a wide range of experiences. I worked on my writing skills because I took a bunch of courses outside of the sciences, and I've always had an interest in that, to be fair. I had the luck that one of my friends said, oh, this person is looking for a volunteer. So I worked in a great lab, and I worked with a good PhD student. Funny enough, footnote here, the supervisor of that lab never spoke to us volunteers, never interacted with us, but this PhD student was a good sponsor and helped me basically get a grad position. So I worked in Sangeeta's lab. It was a small lab, but provided great guidance, strong mentorship, and gave me an opportunity to network. Now here I put this bullet point of visa issues, but that's related to when I was looking for a postdoc. So I was initially supposed to go to the US to conduct a postdoc. Everything was set, but of course at the time I only had an Iranian passport, and that was virtually impossible. So that gave me a 5-month delay in going from my PhD to my postdoc, which was unfortunate. So I've put the blue as unfortunate or bottlenecks, essentially. Again, in Peter's lab, a lot of RAs, a lot of postdocs, and a very fast runway to publications, and the ability for industrial partnerships, and an excellent mentor sponsor. On the flip side, that was 3 years being in Calgary, away from my fiance at the time, and that is something that goes untold. Of course, in this academic trajectory, it's thought that everyone can go through it and go through all of these different places and land in the right place. And that is especially hard, of course, for minorities, but also women. But my spouse, she is a consultant and had the means to come to me and also to basically be the benefactor for me going on my academic escapade. At Brock, again, Dr. DeLuca, excellent mentor sponsor, and helped me train for interviews, helped me think about what I want to do. And without that, it would be a lot harder to land a job at McGill. So there was a lot of luck on the way, and also importantly, good sponsors. So my 4 points to both grads and on the flip side, faculty. So to grad students, you have to persist, but you have to be agile and seek skills, right? So seek stuff that will differentiate you from others. But you have to also stay focused on what you want to achieve. And to faculty, be more than just a mentor, but be a sponsor, be intentional about it, give opportunities equitably, because I see a lot of labs where they see a student who is good at presenting or interacts well and give all the opportunities to them and not the students who are accomplishing the work. So equitable access is important, language is important, and recommendation letters have to be basically it's important the way you write recommendation letters. So there's a lot of research around how women are presented in recommendation letters versus men, and the adjectives that are used there. So be cognizant of that, and read about it. Frankly, it's a learning process for me as well. And providing language, again, is important when you're training students. And finally, just being considerate, thinking of others, and increasing the reach of science and research to your undergrads, to the public at large, etc. So I'll end there. Thank you.
Well, thank you so much for two wonderful presentations. We got a treat today. Great job. I really enjoyed both presentations. And I have a question to kick off the questioning period, and goes to the science part. Like you, I was at the University of Calgary for a while in the 90s. And at that time, plants, we just learned to efficiently engineer plants. And so my PhD work was on producing a fish growth hormone in the seed of canola. And the idea there was to be able to easily mass produce, grow plants and mass produce this recombinant protein and use it in an industrial setting. Now, the question is, you are going now the opposite way. Back then, we argued a lot that how easy it is to grow plants, how we have all the infrastructure set for it, farming is set, we can grow big farms, lots of plants, biomass is a lot, and plants are already there, and we use them for other things, so we can easily produce something using recombinant DNA technology in plants. And so it's all good to use plants as a platform for producing recombinant material, whether it's small molecules like you're working with or proteins. What happened? Why are we back going to microbes? Back then, we would argue all the times that, oh, bugs are so bad for you, and you got antibiotic resistance, and then so expensive to grow in, in fermenters and so forth. And here we are again going back to the microbial systems. What do you think happened?
I think there's a push and pull between these two approaches, right? So while I think maybe in the past 5 or 10 years, there's been a real kind of sea shift back to, as you say, back to microbes, in particular yeast. Of course, nothing is sequenced to the level that the yeast is, and beyond that, protein-protein interactions for every single protein in the yeast proteome has been studied. That you can get a ton of information about all of that. It's tractable. You can produce synthetic genomes that are knock out everything that is irrelevant, so to speak, and you can introduce pathways orthogonally, right? So in plants, there's so much complexity and so many different layers where I would say it's almost impossible to introduce an orthogonal pathway. And by that, I mean something that doesn't affect the other endogenous pathways, something that you have the endogenous pathways, it takes an intermediate and goes basically at a 90-degree angle away from it, but doesn't detract from the other functionings of that cell. But in a plant, that's virtually impossible. So if people can reduce it to the least number of elements, so to speak, where a cell is still sufficient and can basically ferment and grow, and you introduce new pathways, that seems to me much easier than taking a complex plant and hoping that you can introduce a pathway that doesn't detract from everything else going on in that plant. Or maybe you basically introduce a pathway only in a certain section of the plant. But you can agree with me that plants are more complex than bacteria. On the other hand, now there's a shift to single cell green algae or diatoms and introducing pathways there. And the problems there is that you'll get the right type of membranes and the right type of local environment to carry out those reactions. You get the right ER membrane, you get the right type of glycosylation modification of proteins, like the post-translational modifications. So maybe the future is, algae. So I guess that's my answer.
And a related question, thanks for the answer. You mentioned that you're using yeast like many others, yeast as a platform. Now, if you were to think about the yeast as a cell, a eukaryotic cell, and compare it to a plant cell, would you say it's more similar to a mesophyll cell or a cell that you find in glandular trichomes? You mentioned that a lot of times plants produce specialized metabolites in glandular trichomes, which have highly specialized cells that are dedicated to producing these secondary metabolites. Probably it's more so for some terpenes that I look at compared to some alkaloids, but maybe you can, I believe there's a certain level of specialization for alkaloid metabolism as well. But these cells are not as specialized at the very least to say as the secretory cells you find in glandular trichomes. Maybe you can comment on that challenge as well.
That's 100% true. And that gets to kind of the heart of this, the unknown business, right? So when you look at glandular trichomes, you look at, resin ducts in coniferous plants, you look at the lenticifer of opium poppy, you have these metabolite protein, almost like complexes or bodies that form in the cell, and you get this thick mixture that is sometimes called nades, which is natural something eutectic solvent protein mixture. So you basically think of a dried out grape, a raisin. You have sugar at an incredibly high concentration, yet you don't get crystallization, right? So how is that happening? You have these mixture of metabolites and proteins and other things that basically allow massive accumulation of a certain compound or a set of compounds, defying all of our understanding of what a cell is. So can you transform a yeast cell into something like a resin duct or a lenticifer? Probably not. But again, can you do that to a single cell algae as well? Again, questionable. But for example, with like Chlamydomonas, a single cell green algae, you have the pyrenoid, which is that sheath around the chloroplast to concentrate carbon. Can you concentrate alkaloids to have everything function there? These are probably the challenges. So yeah, you're right. I mean, going beyond yeast is maybe the future. But in terms of just functional complementation of work that we can do in vitro or silencing in the plant, I think is still a valuable tool. Yeah.
Excellent. Thank you very much for that nice coverage. I'd like to open the floor to others. Are there any questions from other participants? You can put your question in chat or raise a hand, and Robin will let you or enable you to ask your question.
Shirley has a question.
Hello, Dr. Dastmalchi. Thanks a lot for bringing us an excellent talk, and I really appreciate that you shared your career pathway and valuable experience to our graduate students. You really tackled a lot of challenges to become a prof. I want to ask, what made you choose a career in biology, in biochemistry? Did you have any particularly influential role models when you were a new researcher?
Yeah, it's a tough question. How far back do you kind of go? You go back to your family. Of course, I'm privileged that both of my parents are academic in spirit. And my mother has 2 PhDs different fields, in international law and international law. Did I say law twice? I meant relations and law, 2 different PhDs. And so a bit of an academic spirit in the family. My brother was a pharmacist, so that kind of interest in chemistry kind of blossomed there. But most important, I think, was in the 3rd or 4th year of my undergraduate, where I got exposed to plants as assistant that we can study. I was really fascinated by the plant genetics, where from an embryonic stage, the cells are determined. Their fate is almost determined, and you get that architecture forming. At the globular stage, the heart stage, etc. So that's where my interest for plants grew. But in terms of which exact pathway you take, it kind of depends on which lab you get into first. I was interested in a lab that also did extracellular matrix. Maybe I would be doing cell biology now. So it's a funny thing going down the academic path. You don't necessarily always actively push your career forward yourself.
Thank you.
Robin, go ahead.
I was thinking a lot in your last bit where you were talking about sort of all of those things you listed in gold as the unknowns of an academic trajectory. And as someone who teaches primarily undergraduates, and my focus is teaching undergraduates, I think a lot about how to make those things more transparent and obvious to students, so that the people who don't have the privilege that I also come from, an academically minded family, so that some of that was already known to me, but not everybody's like that. So how do you, or maybe how do we, help students know that those things are things that exist and that they should pursue?
Yeah, so as I said, I teach like an intro plant bio course where we go through everything from from green algae to angiosperms and all that. That's pretty common in every university that I've been at. But the way I start my lectures, and I grandly think of this as inquiry guided learning or what have you. So I start every lecture with a slide that is either a question or an image where I pose something that they can think about as the lecture goes along. Sometimes it's taken from research, and then they get the whole process of discovery kind of embedded in their mind. And hopefully it gets them thinking. So I have a lot of students here who are mostly fated to go into veterinary sciences. When I ask that question in the first lecture, that's what they say. And then they see stuff about plants that they've never thought of, and then they get interested in plants. But more importantly, every lecture I try to put in something about research, to be like, people are still studying this, people are studying Marcantia. You think, what the hell is Marcantia, and what's the point of studying these things? And I say, okay, this is still being studied, the biomechanics of this stuff, splitting dichotomously and growing, so that they know that this is stuff that's happening. Importantly, I always talk about the USRAs at the start of our course. I talk about the honors thesis option, and I mention faculty members that are leaders in their field. I say, these people, you might be taking their courses and you think, who is this person coming here? They don't even have the right fashion sense, but maybe globally they're the leader in lignin or something. And that lignin doesn't mean anything to you, but so I try to kind of embed them in what's going on widely in campus beyond my own research. Yeah.
Awesome, thanks.
If I may, I have another question going back to your science, which I can relate to, because like you, I study specialized metabolism. Now, you talked briefly about transporters and the question of transport within plant cells that produce these valuable compounds. What's new in that sense? There's certainly some knowledge available on transporters with phenylpropanoids or phenolics, but not so much for terpenes and alkaloids. Maybe you can comment on that a little bit.
Yeah, so one of the other projects that I did when I was with Peter was publishing on this class of benzyl alkaloid transporters that we found that is less studied, which is the family of purine permeases. And so these are a large family, and we found a set, I think, of 9 that could transport these opiate alkaloids all the way, actually starting with dopamine transport all the way to something like morphine. And there was a range of different substrates that they accepted, and some were good at one reaction versus the other. But you're right, like, when to my undergrads, I talked about nicotine transport and how it's synthesized in the roots, transported out of root cells into the vasculature, goes up to the leaves, and then has to get from the vasculature into leaf cells, and then subcellular level into the vacuole. So there's a lot of different steps there. And for most alkaloids, we don't know the different ins and outs. And beyond that, what I found really tough is to distinguish between importers and exporters and how you manage that. So that is incredibly tough. In yeast, it's tough. And I know that a lot of people do it in like Xenopus oocytes. But then again, that's not a plant. So I think that's a major challenge as well. Yeah.
What's the status of ABC transporter research?
I don't know. I know that your former student Zary Hu, I think, found an ABC in MIA biosynthesis transport. And yeah, I mean, MIA biosynthesis, that's another one where it goes through 4 different cell types, allegedly, and in and out of different compartments as well. So yeah, that should be another area of focus for people. Yeah. We're not doing that right now, but it should be.
Thank you. Any other questions?
We have one last question in the chat, and then we should probably wrap up.
Robin, do you mind reading the question? I don't
Sure, I can read it. Lauren Erlin posts, first she says, thank you for your talk. One of the things you emphasize is that building relationships at the early career level is really important, but I find this is a lot more challenging for some. Do you have any recommendations or ways to support / reach out to students who might be more nervous or self-conscious that might facilitate this?
Yeah, so this is interesting. Since I've taught this interplant bio course twice, I each time I've put out the call for USRAs, and I've actually gone, 5 or 6 students who reach out by email who are interested in doing it. And then of course I'm limited because, NSERC has very stringent requirements for GPAs. So I can't necessarily hire the students that I want to. But I have found that, trying to be approachable, trying to be available to them, after class. Some students are okay approaching you right after a lecture, and others prefer the email. Others prefer to come to office hours. So, having different avenues out there for them. And maybe it's in the lab setting, if you have an in-person lab and you're going through benches or whatever, and you see students working, being like, oh, that's excellent. Or maybe sometimes the feedback you leave them on an essay. When I see like really good essays that are written, I say, that's fantastic. You could do anything in scientific communication. I leave that as a feedback. So and just give them different options of reaching out. And to universities, allowing for USRAs outside of the NSERC one, where GPA is not the only point of criteria, is good. And that's something I've been pushing for over here at McGill.
Robin, there was one more question. If you don't mind, I'll post it. This was from another one of my students, Sophia Ventosa. She wants to know how to reach you if she wants to have a position in your lab after she graduates.
Yes, email is a fantastic starting point. Yeah. I try to answer every email that at least addresses me by my name and doesn't just say dear professor, because I know that there's a lot of those. And yeah, so I really try my best to answer every single email. So yeah, definitely email me. And thank you, Robin, for putting the link there. Yeah.
Excellent. I think, Robin, you mentioned that our time is up. I'd like to do my part in thanking you, Mehran, for joining us and treating us with two excellent presentations. Thank you so much. And thank you, Robin, and everybody else to giving me the opportunity to be the host of Dr. Dastmalchi. I haven't seen him for quite some time, so this is a treat in many ways. With that, I'll pass it on to you to wrap up the session. Thank you.
Yeah, I don't have anything to add. Shirley just put in the chat our upcoming talks. So we have another talk next week. We have one every week till the end of April. And we would love to see all of you back. Thank you so much, Dr. Dastmalchi.
Thank you. Thanks again for inviting me. And thanks to all of you for setting this up, Shirley, Kat, and yourself, and Sohail for arranging this and making it possible. Thank you.
Thank you, Mehran. Have a nice day. Bye, everybody. Nice to see you.